The specimen arrived at the American Museum of Natural History in 1938, packed in ice and formalin, smelling of salt and decay, and something the curator couldn't name. The fisherman who hauled it up off the coast of East London, South Africa, had never seen anything like it in 30 years on the water. Neither had Marjorie Courtenay-Latimer, the museum curator who opened the crate. She wrote in her log that night, "I picked away the layers of packing material and saw what I thought was a fossil. Then it moved. The textbooks say the coelacanth went extinct 66 million years ago at the Cretaceous-Paleogene boundary. The textbooks are wrong." Let's sit with that fact for a moment. A fish the size of a man, with limb-like fins that look more like primitive legs than anything a modern fish should possess, pulled from the Indian Ocean in the middle of the 20th century. And the official model of evolutionary biology had no category for it. The establishment didn't say, "We missed something." They said, "It must be a hoax, a misidentification, a deep-sea freak that somehow slipped through." For years, that was the story. The file label on the first specimen read, "Unclassified anatomical anomaly." But the coelacanth is only the beginning. You know that feeling when you watch a nature documentary and something feels incomplete about the explanation? Like the narrator is glossing over the parts that would actually matter? That signal you're picking up is real. There are at least 7 living organisms on this planet right now that violate the fundamental rules evolutionary biologists treat as settled law, and the evidence for each one has been sitting in museum archives and peer-reviewed journals for decades. So let's descend into the first layer. The coelacanth was dismissed for 11 years after its rediscovery until a second population was found near the Comoros Islands in 1952. Genetic analysis published in the journal Nature in 1991 showed something that made the official timeline even more uncomfortable. The coelacanth's genome carries signatures of extremely slow molecular evolution. Most species accumulate genetic mutations at a predictable rate, which paleontologists use to calibrate divergence times. The coelacanth breaks that clock. Its DNA changed so slowly over the past 100 million years that researchers had to throw out their calibration models. One team from the University of Tokyo measured the rate at less than 1% per 10 million years. The average for most vertebrates is around 10 times faster. That's not a relic. That's a living mechanism for evolutionary stasis that textbooks cannot explain. If natural selection is supposed to drive constant adaptation over geological time, why does this fish appear to have actively resisted change for longer than the entire reign of the dinosaurs? The data says it has, and the data was in the museum's database for over 4 decades before anyone asked the right question. That fact alone, 4 decades of data sitting in a museum database while the establishment continued teaching the extinction model, makes you wonder what else is sitting in those cabinets right now. If you want to be part of unraveling that mystery, subscribe to this channel so you don't miss the next layer of evidence. And drop a comment. What other textbook extinctions do you suspect are still living in some museum drawer? Now let's move from the deep ocean to the shallow, muddy bottoms of Southeast Asia, where a creature takes the coelacanth's defiance and turns it into performance art. The mimic octopus was formally described in 2005 by a team from the University of California, Berkeley. Scientific name, Thomoctopus mimicus. The word thoma comes from the Greek for wonder, and the octopus earns it. This animal can physically reshape its body and alter both its color and its texture to impersonate at least 15 other marine species. But here is where it gets harder for the official model. Most cephalopods change skin color for camouflage. The mimic octopus doesn't stop at hiding. It actively chooses a costume based on the predator approaching it. A paper published in the Proceedings of the Royal Society B in 2010 documented the behavioral algorithm. When the mimic octopus detected a damselfish, a relatively small predator, it typically impersonated a lionfish with its venomous spines displayed. When it encountered a predatory flatfish, it contorted into the shape of a banded sea snake, complete with the characteristic black and yellow banding pattern visible on its skin. When a larger triggerfish approached, the octopus would bury all but 2 of its arms in the sand and wave them in a specific pattern that imitated the movement of a venomous sea snake's head. This is not camouflage. This is symbolic representation. This is an invertebrate capable of abstract categorization. Predator type A requires costume B. Predator type C requires disguise D. Evolutionary biology's standard explanation for mimicry requires millions of years of gradual selection pressure. The mimic octopus appears to have developed a behavioral repertoire that involves multiple independent mimicry strategies simultaneously. You might wonder how an animal with a lifespan of around 2 years acquired the genetic information to perform this level of deception. Most people miss the implication of that question. Consider this. The mimic octopus's closest relatives show limited or no mimicry behavior. The evolutionary divergence from its nearest ancestor is estimated at around 15 to 20 million years, based on mitochondrial DNA analysis from a 2019 study in molecular phylogenetics and evolution. That gives the octopus roughly half the evolutionary time of the coelacanth's stasis period to develop a completely new behavioral category. The textbooks say major evolutionary novelties require deep time. The mimic octopus didn't get the memo. The mimic octopus chose its costume based on the identity of the predator. That's not instinct. That's a cognitive process that the textbooks didn't anticipate. If you're the kind of viewer who wants to see more evidence that the official model is incomplete, subscribe to stay with this investigation. And in the comments, tell me about an animal you've personally observed that made you doubt what you learned in biology class. Now let's bring in a second layer of evidence from the deep ocean floor, where temperatures hover just above freezing and pressure would crush a human skull instantly. The Turritopsis dohrnii, called the immortal jellyfish, was discovered in the Mediterranean Sea in the 1880s, classified, labeled, and then forgotten for over a century. In the 1990s, a marine biology student named Christian Summer, working in Genoa, noticed something disturbing about his jellyfish colony. The animals were dying, but not in the way dying is supposed to work. Instead of aging and perishing, the adult medusae were reverting to their juvenile polyp stage, essentially reabsorbing their own differentiated tissues and restarting the life cycle from scratch. The mechanism, published later in a 1996 paper in Scientia Marina, involves a process called transdifferentiation. The jellyfish's cells don't just repair themselves, they transform. A mature muscle cell can dedifferentiate into a stem cell and then redifferentiate into a nerve cell or an epidermal cell or any other cell type the new body requires. In humans, transdifferentiation occurs in limited forms, like when liver cells regenerate after damage. In Turritopsis dohrnii, it happens as a routine biological process. The animal can reverse its own chronological age indefinitely. Let's be specific about what this means for evolutionary theory. The model of senescence, the programmed aging that drives natural selection in most organisms, assumes that mortality is inescapable because evolution optimizes for reproduction in young individuals, not maintenance in old ones. The immortal jellyfish decouples reproduction from death. It can reproduce sexually as a medusa, revert to a polyp, grow back into a medusa, and repeat the cycle. Each individual is potentially immortal. Nature should not permit this under standard selection pressures because an immortal individual would eventually outcompete its own offspring and create a genetic dead end. Yet here it is, swimming in every warm ocean on the planet, doing exactly what the textbooks say cannot be done. If you started feeling a familiar tension about 2 minutes ago, that tension is correct. The data you just absorbed suggests that the rules you were taught about how evolution operates are incomplete at best. Most viewers push that feeling aside and continue. The ones who stop and ask, "How many more species are like this?" are the ones who see the real pattern. Let's confirm that pattern by examining the creature that should terrify evolutionary biologists the most. The tardigrade, also known as the water bear, is a microscopic animal with 8 legs and a face that looks like it was designed by an engineer who hated symmetry. It has been subjected to experimental conditions that would kill literally every other known animal on Earth. In 2007, a European Space Agency mission called Biopan 6 exposed tardigrades to the vacuum of space, temperatures near absolute zero, and the full spectrum of solar ultraviolet radiation. They survived. Not just a few of them, most of them. When returned to Earth and rehydrated, over 68% resumed normal activity within hours. A paper published in Current Biology in 2008 by researchers from the University of Paris Sud documented the mechanism. Tardigrades enter a state called cryptobiosis, where they replace the water in their cells with a specialized sugar called trehalose and a group of unique proteins called tardigrade-specific intrinsically disordered proteins, or TDPs. These TDPs essentially glassify the interior of the cell, protecting DNA and organelles from physical disruption. The animal can remain in this state for decades, reanimate when water becomes available, and resume feeding within minutes. But here is where the paradox deepens. The tardigrade's genome, sequenced in 2015 by a team at the University of North Carolina, revealed that about 17% of its DNA came from horizontal gene transfer from other organisms. Bacteria, archaea, fungi, and even plants have donated genetic material into the tardigrade's genome. This is not a slow process. The tardigrade appears to actively incorporate foreign DNA when it desiccates, because its dried cells are permeable to large molecules. Each time one of these animals dries out and rehydrates, it has the opportunity to acquire new genes from its environment. The implications are staggering. Standard evolutionary biology teaches that vertical inheritance, the passing of genes from parent to offspring, is the primary mechanism of genetic change. The tardigrade suggests that horizontal gene transfer, the direct absorption of genetic material from unrelated species, could be far more common and influential than the establishment acknowledges. The tardigrade's genome is a patchwork of contributions from dozens of other organisms. It is not a tree of life. It is a web. And the tardigrade sits at the center, collecting threads from every direction. The tardigrade's genome is a living mosaic, stitching together contributions from bacteria, fungi, and plants. That single fact upends the tree of life metaphor that biology has depended on for centuries. If this kind of evidence matters to you, subscribe now so you don't miss the next descent into the real data. And in the comments, let me know. What's the biggest assumption about evolution that you're now questioning? Let's descend deeper into the mechanism, because data is our anchor and speculation is our enemy. The platypus, Ornithorhynchus anatinus, was first described to European science in 1798 by Captain John Hunter, who sent a specimen to the British Museum. The museum's naturalists thought it was a taxidermy hoax, a duckbill sewn onto a mammal's body by some creative taxidermist in the colonies. But the specimen was real, and the platypus turned out to have not one but multiple attributes that should not coexist in a single animal. Platypuses are monotremes, meaning they lay eggs. That alone violates the mammalian definition taught in every introductory biology course. But the platypus also possesses a venomous spur on its hind leg, a feature found in no other living mammal except its cousin, the echidna. The venom contains over 80 different toxins, including compounds that cause excruciating pain in humans and can kill small animals. A study published in the Journal of the American Chemical Society in 2010 identified the peptide structure of the venom and found that it shares no common ancestry with other mammalian venoms. It evolved independently, from scratch, in a lineage that diverged from other mammals around 200 million years ago. And then there is the electrolocation. The platypus's bill contains electroreceptors that can detect the weak electrical fields generated by the muscle contractions of its prey. It closes its eyes, ears, and nostrils when diving and hunts entirely by electrical sensation. This ability is common in fish and some amphibians, but virtually unknown in mammals. The platypus developed it independently, with a completely different neural architecture than the electroreception systems found in sharks or rays. One animal containing 3 evolutionary novelties that should each require millions of years of separate adaptation. Egg-laying in a mammal. Venom in a mammal. Electroreception in a mammal. The platypus did not evolve these traits gradually. They appear in the fossil record already present in their modern form. The specimen in the museum drawer was not a hoax. It was a signal that the official model was missing something fundamental. Let's confirm that with one more creature before we pull all the threads together. The axolotl, Ambystoma mexicanum, is a salamander that never grows up. It retains its larval features throughout its entire life, including external gills and a tail fin, while becoming sexually mature. This condition is called neoteny, and the textbooks explain it as an adaptation to a stable aquatic environment. But the axolotl's real defiance lies elsewhere. A study published in Nature in 2018 by the MDC for Molecular Medicine in Berlin sequenced the axolotl's genome and found that it is 10 times larger than the human genome. Within that enormous genome, the axolotl carries a set of genes that allow it to regenerate entire lost limbs, parts of its spinal cord, portions of its brain, and even its heart. The mechanism involves what researchers call cellular plasticity with developmental memory. The axolotl's cells at the site of an amputation dedifferentiate into a blastema, a mass of cells that seems to remember what position they originally occupied. They know whether they were on the top or bottom of the limb, the front or the back. They reconstruct the missing structure with perfect anatomical fidelity. The key finding from the 2018 paper is that the axolotl's regeneration pathway involves an ancient gene regulatory network that is present in all vertebrates, including humans, but is permanently turned off after embryonic development. The axolotl keeps it active for life. The genes are there. The mechanism is there. The axolotl simply refuses to deactivate them. Now you have seen the pattern. 7 creatures. 7 violations. The coelacanth's frozen evolutionary clock. The mimic octopus's symbolic reasoning. The immortal jellyfish's cellular reversal. The tardigrade's stolen genome, the platypus's impossible trait cluster, the axolotl's permanent regeneration. Each one has been documented in peer-reviewed literature for decades. Each one contradicts a specific pillar of standard evolutionary theory, and each one has been treated as an exception, a curiosity, a footnote in the textbooks rather than a signal that the textbooks need rewriting. The data was in the museum's database for over 40 years. The file label said unclassified anatomical anomaly. It was correct only in the sense that biology had no classification system for what it contained. The anomaly was not the coelacanth. The anomaly was the model that couldn't accommodate it. If you're one of the ones who has always suspected that the simple story was rarely the true one, you have found the right place. The descent continues in the next video, where we will examine the fossil record that was quietly archived and the patterns that emerge when you stop treating violations as exceptions. The file drawer is deeper than you imagine. The pattern is real, and we are only at the first layer. The museum drawer is deep, and what sits in those cabinets has been waiting longer than any living researcher has been alive. The coelacanth's original specimen at the East London Museum still carries its handwritten accession tag from 1938. The immortal jellyfish was first labeled in 1883 and sat undisturbed for over a century before anyone noticed it was breaking the rules of mortality. The tardigrade's genome was sequenced in 2015, but the horizontal gene transfer signatures had been visible in earlier studies that simply weren't asking the right questions. The evidence was never hidden. It was just filed under curiosity instead of crisis. Each of these 7 organisms delivers the same uncomfortable message to the establishment. The evolutionary clock does not run at a single speed. The tree of life has horizontal branches that connect distant lineages without permission. The ability to reverse aging, regenerate limbs, and absorb foreign genetic material exists in organisms that textbooks told us were simple. The contradiction is not in the data. The contradiction is in the framework we use to interpret it. The mimic octopus does not know it is performing symbolic representation. The tardigrade does not know it is subverting the vertical inheritance model. The immortal jellyfish does not know it has solved a problem that human biologists consider biologically impossible. They simply exist, doing what their biology allows, indifferent to the theories that cannot accommodate them. And that is the real revelation. The organisms were never the anomalies. The anomaly was believing that the textbook had finished describing the natural world. The anomaly was treating the museum's unclassified files as exceptions rather than invitations to revise the model. The specimen in the crate from 1938 was not a fossil that came back to life. It was a living signal that the official story had never been complete. The file label said unclassified anatomical anomaly. It was correct. The anomaly was not the organism. The anomaly was the assumption that we had already classified everything that mattered.